Fantastic. Okay, so today I'm going to present this mainly this paper, to be honest, that is more methodology than proper results, since I'm a biomedical engineering, not a medical doctor. So first of all, let me to thank my collaborators that mainly works also in Rome, in the Department of Neuroscience and Rehabilitation in San Raffaele of Rome, in Italy. So since the title is like dynamics of the cognitive brainwave P3b at rest for Alzheimer's dementia prediction, mild cognitive impairment, I would like to introduce first of all, just in case some of the audience is not aware about, the idea of, very quickly, of course, the idea of the subcomponent linked to the P300, that's the P3A and the P3b. And after, I'm going to show you or introduce a little part of this method or algorithm that is called functional separation, and in which way we were able to use the potentiality of this algorithm to enhance the P3b for prediction in from mild cognitive impairment to Alzheimer's dementia. And this is the last part that is mainly the title of this paper. So let's start with some background then. The P300 is an event rate potential that has been heavily investigated for as a neural marker for attention and cognition. We can evoke the P300 in many different ways, so I suggest for people that are more interested in going deep about the P300 to have a look to this nice review done by published in 2007 about the P300. Very nice. I took some picture, some figure from this review, just to introduce the idea of how we can evoke the P300. The single time stimulus is based on a target response that give us evoked activity around 300 milliseconds, slightly later, 330, 350 milliseconds. After we can analyze the P300 using a notable task that mainly is standard use, like a standard stimulation with target stimulation, in which the target stimulation gave us the P300. Instead, if we want to evoke the two components of the P300, it has the P3A and P3B, as we did in our task, we have to use also a distractor stimulus. So we can use a notable task plus a distractor stimulus. And as you see, this three stimulus model is evoked the P3A through the distractor, instead the P3B through the target. As you see, but we'll see later on that there is many different processes that undergo through these two evoked response. In fact, if we can see from this preliminary results, or just sorry, it's not preliminary results, are very consolidated results that are very well in the literature, that we have for the P3A a more frontal topography, and for the P3B a more parietal topography. From this, we can understand that we have different neural sources that underpinning the P3A and the P3B. We can also have a look at these two papers that discuss very well this property and about the generator of the P3A and P3B. But also from the morphology, since we are going, since we are going to see the morphology of the P3B in this case, and the P3A in this case, that I superimpose in this new part of the figure, that give us the idea that the P3A is relatively consistent across trial, and in fact you have a very clear evoked response, and very peaked around 380 milliseconds in this case. Instead, for the P3B, since it's a response locked, it changes slightly from trial to trial because, of course, depend on the speed of the response. So the P3B is the more cognitive one. Consequently, we can say and we can confirm what is already well established about these two papers and many others, to be honest, that this kind of P3b has a widespread morphology that ranging from 300 to 6 until 600 milliseconds, and is strictly dependent also from the cognitive performance of the subjects and also the age of the subjects. This difference in scalp topography and ERP morphology reinforces the idea that the P3b might serve different cognitive process. And this is what we try to do and to verify using our functional separation and method or algorithm, and with the idea to answer this two, let's say, question or aims of the paper, that is one channel enough to describe the brain process underlying the P3A and P3b components? This is the first question that we try to answer. And the second question is, the P3b neural dynamics are more sensitive marker for AD prediction in MCI. So let's start with the first question, but before I'd like to just save a couple of minutes, of probably 1 minute on the functional separation, because otherwise we are not able to follow the next part of the presentation. So I make a comparison with ICA that probably is more aware as an algorithm. ICA is part of the blind source separation method, and also functional source separation with a slight little difference between ICA, in which we assume as an hypothesis that the mixing system, so how we record the data through EEG on the scalp of our head, are linear and is time invariant. And for the two algorithms, the hypotheses on the mixing system are exactly the same. So it's as an ICA approach, a clear ICA approach. What is changing between the two algorithms is the hypothesis on the sources. Then hypothesis in the ICA, so independent component analysis, the sources are statistically independent. Instead, in our functional separation, we add a different constraint. So this hypothesis all the hypotheses needed for resolving this equation, since we have two unknown and one equation. So to resolve this equation, we need more hypotheses. Otherwise, we have more unknown than equation. And for the ICA, just to remark that we use the sources are statistically independent. Instead, for the functional separation, we use some functional constraint that come from the data. What gave us this hypothesis more respect to the ICA is that instead of have many sources, we can have just one source that represent totally the hypothesis of our function constraints. So now the question is, what means functional constraint? And functional constraint is something that coming from our data. In this particular case, we maximize, instead of the independency, we maximize the activity around 300 milliseconds. In this particular case, around 380 milliseconds. This is the question that we are going to use for maximizing this. So forget about the question. It is more the idea that is important to pass in this talk. The idea is that we maximize our windows that we are interested in particular, this case, the P300. So we have back, let's say, not a source, because we didn't resolve let's say inverse problem, but we have, it's better to call is like a virtual electrodes that is a linear combination of all the electrodes that represent this topography. So what we have this response. This is the response due to a virtual electrodes. As you see, for just check how well we were able to extract the source around 300 milliseconds, if we do a discrepancy, so a difference between these two conditions, we can see that all the activity that around 300 milliseconds are in our virtual electrodes. So if this is clear, we can pass to the idea how we evoke the P3A, P3b, and we use three stimulus visual oddball task with 19 EEG channels. And this is another choice that we done because we want to be in a pure clinical environment. So we didn't use fancy EEG with 156 channel because, to be honest, in this kind of literature, if you see if they use a very sophisticated system with huge number of channel, in the end, they are going to use a selection of the channel that mainly are CZ, PZ, or FZ. So we have to understand why we have to use all this channel if to the end we are picking just some channel. For in this condition, instead, we are going to use a 19 EEG channel that is a clinical condition that all the hospital has, and they are using in the standard clinical situation. Each stimulus is around are very standard for visual oddball tasks. Which are our tasks? Our tasks are standard stimulus that is a purple circle around 3.5 centimeters and appear for 80% of the trials. After we have the target that lies the P3a, that is a slightly different purple circle that is 4 centimeters instead of 3.5 centimeters and appear for 10% of the trial. And after, as I introduced before, we have a distractor stimuli, that is a completely different one, that is a checkboard, a black-white checkboard that appears also for 10% of the trials and lies the P3a. Let's get a look about our cohort of the subjects. So we recruit 175 volunteers divided in this way. We divide young population, 20 young healthy subject, and 20 elderly control without any cognitive deficit. And these two groups were mainly used for extracting the spatial filter, so the topography that we saw before, and to compare with the elderly population, just to check if our algorithm works well in extracting our components or our P3a and P3b. Afterwards, we use the MCI, S means stable, that do not convert to Alzheimer after the follow-up, and converted one that convert in after the follow-up. So we use the information and the topography. This is the reason why I am also happy to call this approach like a spatial filter to apply to the MCI for extracting the component of the P3a, P3b in many different conditions. Just this is another verification how well works our algorithms. And as you see, if we compare elderly population with young population, we have a very nice peak around 380 milliseconds for the young, a slightly slow one, P300, in particular this P3a for the elderly. And that this is just to demonstrate what is very clear in the literature. And also for the P3b, what we demonstrate that the amplitude of the P3b is a little bit less power respect to the young population. And this is guarantee what we found is a standard finding that is clearly showed in the literature already for more than 30 or 40 years. If you like this idea of approaching, I suggest this paper that I did in collaboration with the ATH in Zurich, where we demonstrate already that the P3b amplitude is a nice marker for cognitive decline in older population. But in that case, was not Alzheimer or MCI population, but just was older population. Now let me explain this slide because I understand that it's a little bit complicated, because this is what I'm trying to push as a new methodology, and probably some of you are interested in it. So we use evoked response or a recording session for audible task with the P300 stimulation, with the aim to extract through the functional separation our two spatial filters, one for the P3A and one for the P3b. Once we have obtained the P3A and P3b, remember the functional separation is, as the ICA linear method, then we can invert these two spatial filters, and we use these two spatial filters to extract the activity in a completely different condition. In this case, we are going to use a rest condition with open eyes. Then what we have with this spatial filter applied to a rest condition, we have a virtual electrodes that represent the best combination of all these electrodes for P3A and P3b, but with just one time series that maximize this idea of the P3A or P3b. Then, thanks to the functional separation, we are able to characterize the dynamics of this virtual electrode for example, to test if these are a marker for MCI that predict AD. In which way? So we test, just to reinforce this idea, what is mainly used in the literature. So we choose the channel and we compare channel across our virtual electrodes. So instead of using one channel, we use the combination of all the channels. And we apply a repeated measure ANOVA, in which we use groups. For groups, we use MCA stable, converted, and elderly condition. And bands for us a repeated measure, in which we divide the activity FZ, CZ, PZ, and our virtual electrodes coming from P3A or P3b in delta, theta, alpha, beta, and gamma. What we found from this repeated measure ANOVA, we found it was an indirect effect and a between subject factor that show difference among groups, and this is the significance. And in particular, this is the post-hoc test, shows that we have difference between stable and converted MCA in delta band and in theta band, but not for the other band. That is something that is not surprising. This is already known in the literature. So the problem that is mainly in the literature, sometimes they don't use this repeated ANOVA because are very restrictive from the statistical point of view. And normally they isolate the bands like delta or theta, and they use a simple ANOVA. But I mean, the correct way to proceed is using a repeated measure ANOVA, because this is a repeated measure of the same group of the subjects. In fact, if we approach with the repeated measure ANOVA, we didn't have any significance for the P3A special filter and for the channel selected. In a way, we reinforce the idea of what we found already, that the P3b is the more sensitive marker for cognitive decline. To answer a little bit more in deep this idea, is one channel enough to describe the brain process underlying the P3A and P3b? What we did, we approach with, let's say, a Bayesian statistical analysis, in which we did a t-test, but with the Bayesian factors, for the MCA stable and converted, for the P3b, FZ, CZ, PZ. For the theta band, because we found that the theta band is the one that survived to this test. So we found that for P3b we have significance. We have also significance for FZ, CZ, and PZ. But what we found, and what gave us the Bayesian factor and this kind of approach, we can test the quality of the marker respect to the other markers. In this case, the Bayesian factor was 1,700, let's say 1,800 times, and instead for the best electrode, that is the PZ, is 13. This means that if we compare the Bayesian factor, we found the spatial filter that used the FS P3b dynamics was 138 times more sensitive to detect difference between MCI stable from MCI converted. To go beyond the idea of group level analysis, since we want to find a marker that is able to predict this conversion, we use, and then answering this second question, so is the P3b neural dynamics a more sensitive marker for AD prediction MCI? We use a machine learning approach, in particular we use linear discriminant analysis, for what? For discriminating between stable and converted and elderly control. The results, I'm a little bit long, but it's almost done. The more important results are MCI versus elderly control, where we reach for the P3b 82.6% of the accuracy, and instead for the best electrodes we reach the chance level, so more or less than 47%, even if the other channel reach 58%. To be noted that if we increase the number of information, for example, using all the electrodes, for example, FZ, CZ, PZ, this do not increase a lot the percentage of the accuracy. And this was the same also if we use FP3b and FP3A. Sorry, here I have an error. This is P3A plus P3b. Then we reach the 79%. So this means that the P3b is what really discriminate. Instead, if we use or we add the information of the P3a, is like something that make the model a little bit more complex and reach less accuracy, even if the accuracy is more or less 80%. The other thing that we have obtained, despite this, that is a spurious idea, this is something that the reviewer asked to add in this table, but I don't believe that this is something that is informative, because this should need to have already this classification. Instead, the idea is that we have MCA and we want to understand if they are converted. So the idea is to make the test between stable and converted, and we got the 64.4% of the accuracy respect this 58% if we use just one electrode. Just I'm going to conclude what we can say in conclusion, that combining the activity from all the electrodes such as FSS or other algorithms that are able to do so might be a good choice to overcome possible misleading results that coming from a single electrodes. And the reason for this is because mainly the P3A, P3b is a very distributed activity, is more a network, because we are already in a cognitive process that is not can be detected just from one electrode or a group of electrodes. And then we need to understand which is the best combination that give us the activity underlying these two processes. And from the Bayesian factor, we say that the spatial filter, especially the P3b spatial filter, gave us a sensitivity of 138 times better than a standard electrode that is normally used in the literature. And from the prediction point of view, so if we are going to use this idea as a marker for prediction, we can say that we are able to predict with the spatial filter P3b around 74.4% of the accuracy. That I understand that from the point of view of the medical doctor, 64% is not a huge, a huge results, but this is what is normally found in the literature, something also a little bit improved if we are going to use only the electrical activity. But of course, if we are going to add other, let's say, predictor, like the test or other marker, maybe we can improve this prediction. This I want to remark to stress the idea that this 64.4% of accuracy is related just with the electrical activity that is normally done in the clinical situation with a rest condition with open eyes. Thank you for your attention.
Thank you so much. That was amazing.